Hey everybody, my name's Emma Griffiths and I'm a research associate at the University of British Columbia in Vancouver, Canada. And I'd like to start by thanking the conference organizers for putting together such a great meeting. I really enjoyed it so far. And for enabling me to tell you a little bit about our work on developing a SARS-CoV-2 genomics metadata standard and a tool to enable public health labs and researchers better harmonize and integrate information across data sets and databases as they continue working towards an increased understanding of SARS-CoV-2 epidemiology and biology. The sequencing and sharing of SARS-CoV-2 genomes with public databases like GISAID and the INSDC has had enormous benefits during the pandemic, from tracking the spread of the disease throughout the world, within countries, and during particular outbreaks, to providing genomic information for the design of quantitative RT-PCR diagnostic tests, as well as vaccines, to refuting misinformation about the origins of the virus. Now, when we talk about public health genomics data, we don't just mean the sequence data. We're also talking about the contextual data. And by contextual data, I mean the sample metadata, as well as the lab, epidemiological, clinical, and methods information that are critical for interpreting the sequence data and making informed decisions based on analytical results. And it's the contextual data combined with the sequence data that enables us to characterize lineages and clusters, identify variants with clinical significance, and correlate genomic trends with outcomes and risk factors. Now, getting the right information to the right people is absolutely critical in a public health emergency, whether it's via data sharing within an organization, between trusted public health partners, with international organizations like the WHO, or with public repositories. But sharing can be difficult when contextual data is encoded in different ways. For example, on the left-hand side of the slide, you can see excerpts of case report forms used by two different public health agencies right here in Canada. And for those of you who are unfamiliar with them, case report forms are the forms a healthcare worker fills in to collect patient information when a patient goes to a testing center for a COVID test. While both forms describe specimens that are collected for testing, they describe them at different levels of granularity. They use different terms, they use abbreviations, and while a human can probably figure out how the information is related, a computer can't unless you tell it exactly how. And these issues are perpetuated from organization-specific databases out to public databases where metadata standards are largely broad to encompass a lot of different use cases and pathogens, and users are left to apply them as they see fit. As an example, on the right-hand side of the slide, you can see two different contextual data records from the same public repository where information is encoded using different templates and standards. So the fields are different, there's a lot of free text, and there's information like latitude and longitude and age and gender that in some circumstances could be considered re-identifiable. While I'm sure that they're fine here, in other cases, they could be considered as putting patient privacy at risk. So the take-home message here, which I'm sure will be no surprise to any of you listening, is that using different data structures makes information less interoperable and more difficult to integrate, which means you need to spend more time and resources to clean it and restructure it before you can use it. The Canadian COVID-19 Genomics Network, otherwise known as CanCoGen, is an example of a national sequencing effort with the goal of integrating viral genomics data from across the country to perform public health surveillance, as well as integrating human genomics data with that viral data to better understand pathogen-host interactions, patient outcomes, risk factors, and to develop therapeutics and improve diagnostics. Now, what you need to know is that Canada is a country made up of 10 provinces and 3 territories. It has a decentralized health system in which healthcare is under the jurisdiction of the provinces and the territories. So, harmonizing and integrating all this information across different databases and different data sets, and critically across different jurisdictions, is not at all trivial. To help us structure all of this contextual information, we created a data specification with an emphasis on public health needs and interoperability in collaboration with the Public Health Alliance for Genomic Epidemiology, better known as PHAGE. And the specification provides standardized fields and terms for describing things like identifiers, sample collection and processing information, host information, sequencing and bioinformatics methods, as well as quality control metrics, pathogen diagnostic testing details, and provenance and attribution, so that all data generators are acknowledged for their contributions. The vocabulary for the standard was sourced from a variety of different case report forms, as well as public surveillance and repository requirements, existing metadata standards, and ongoing advancements in the literature. To highlight where our standard overlaps with existing standards and where the gaps were, we carried out a mapping exercise to a number of existing standards. And currently, we're mapping all of our vocabulary to existing OBO Foundry ontologies. The standard was recently published as a preprint, and there are already a number of early adopters. For example, the South African National Bioinformatics Institute, and they're integrating the spec into their Baobab LIMS, or Lab Information Management System. There's also AUSTRACA, and that's a pathogen genomics data sharing platform used in Australia. There is the American National Sequencing Effort for SARS-CoV-2 called SPHERES, and of course, Cancogen. Now, it's one thing to develop a standard, but it's another thing to enable public health workers to actually start using it. So we also built a harmonization tool called the DataHarmonizer, with the goal of enabling users to harmonize, validate, and work with their contextual data in a private and secure way, using vocabulary that they're familiar with, and to structure this information so that it's more interoperable and more easily reused for different kinds of analyses. Okay, so now is the demo portion of the show. So the first thing a user will need to do is to go to our public health bioinformatics GitHub site and to download the zip file containing the main.html file, which will launch the application. So there you go, easy peasy. The Data Harmonizer is essentially a spreadsheet editor application. So just like you can do a lot of the things that you can do with a regular spreadsheet, that contains all of the fields and terms from our specification. And it's fully functional offline for data security purposes. So your data never has to touch the internet. So you can see here in the Data Harmonizer that there is some color coding. The yellow fields are considered required, and these are types of information that are needed for public health surveillance. There's also purple fields, those are considered recommended, and there are fields in white, and those are optional. So to minimize data entry requirements, users can go to the Settings tab and pull up the minimal required set of fields by clicking on the Show Required Fields tab. To get users on their way, there's a handy Getting Started. There's a help button with a handy Getting Started tab, and this has little videos that show users how to interact with the interface. There's also an SOP that provides step-by-step instructions for entering data and for validation. And there's also a field level reference guide that users can also access by double-clicking on any header, and that provides definitions of the field, guidance for populating it, examples of usage, and what to do if you can't supply the required information. Some of the fields allow more flexibility and free text. Other fields, like those with dates, provide some formatting. There's also pick lists. There's also drop downs with pick lists of standardized terms. There are some fields that provide automated binning. Hope no one gets motion sickness from me flicking through this so quickly. There are also other fields that provide search and autofill. Okay, so for the purposes of the demonstration, let's open some mock data that I created previously. So once a user has entered all their data, they can perform validation at the click of a button. So you just go to validate, and then any errors are highlighted in red. And a user can quickly fix the errors. Then revalidate. And then upon validation, they can either save their work or they can export it to a number of different formats. And currently you can see the available formats are in GIS8 format. So this will prepare submissions for the GIS8 database. There are a couple of different formats for Canadian specific platforms, and coming soon will be a format for NCBI biosample. Okay, so that ends the demonstration portion. All right, so in summary, the DataHarmonizer implements the Cancogen phage SARS-CoV-2 contextual data specification, and it's a public health tool that can be used by any lab that wants to implement the standard. It's easy to install, and it enables validation and easy transformation into public repository submissions. You can find the DataHarmonizer and our preprint at these links. Please feel free to contact us with any question at any time. And finally, I'd just like to acknowledge all of the excellent and critical contributions of Damion, Dooley, Ivan Gill, Rhiannon Cameron, and Will Hsiao, as well as the rest of the Hsiao Lab and the phage Consortium. And thank you all very much for listening.